Nervous system, raise your hand. Immune system, raise your hand. I don't care, raise your hand. I'm just here for the good time.
I just want to learn some shit I don't know.
All right, so it will be nervous system material on Thursday.
Cameraman.
All right, so what I'm going to do today is, no matter how long it takes, I'm going to go over both the light-dependent and the light-independent reactions of photosynthesis. And then if you've had enough at some point, just quietly leave. Okay?
Just leave.
Because I don't know if it's going to take me an hour, hour and a half, or 6 hours. But if it takes 6 hours, then Gemma and I are going to sit here, she and I, and we're going to hammer this stuff out. All right. So, big picture stuff. Here is a chloroplast. Here are the individual little sacs, flattened sacs within the chloroplast, and there typically are many of them, many more than I care to draw. Occasionally, for whatever reason, they have some membrane-like connections between them. I don't know what they're called, and I don't know what they're for, so apparently it's not very important. One sac, one individual sac, is a thylakoid, and an entire stack of thylakoids is singularly a granum and plural grana. And then there's a fluid inside the thylakoid, and that fluid's going to be important. It'll show up in, in a much larger perspective when we start talking about the light-dependent reactions. But there's also a fluid that permeates this whole area that is called the stroma. As a general rule of thumb, it's the thylakoids that are responsible for the light-dependent reactions, the reactions that directly use sunlight. And it's within the stroma where the light-independent reactions occur. And those light-independent reactions are better known as the Calvin cycle. So, again, as a big picture thing, because it's real easy when you do some of this to just sort of get lost. Well, what is going on? You know, the details are accumulating, but I'm not seeing the big picture. As the big picture, the light-dependent reactions are going to be those where the energy of light, typically sunlight, is going to undergo some chemical transformations using photosynthetic pigments. And those pigments are going to help take boosted electrons. Another way of saying boosted electrons is high energy electrons, electrons that are at a energy level that is artificially high for that particular electron. And it'll take those boosted electrons and use the energy of those to make two molecules, ATP, and another one called NADPH. And those molecules, as always, are not made from scratch. They can be made from scratch, but it's pretty energy expensive to do that. So almost always when an ATP molecule is being made, it's being made by an ADP and a phosphate group being transformed into an ATP molecule. And NADPH is an NADP plus molecule plus 2 electrons plus a hydrogen ion. And the energy from originally the sunlight is being used to put those 2 electrons and a hydrogen onto the NADP plus to make NADPH. So in both cases, this ATP and NADPH are the products of the light-dependent reaction. You don't get anything more from it than that. But the whole idea of photosynthesis is to make glucose, or at least make some kind of sugar. And classically, it is most often described by describing it as glucose. So what ATP and NADPH are being used for is to fuel the light-independent reactions. I'm not going to look at the details of how that occurs right now, but it involves a series of reactions that are called fixing, or the fixation of carbon dioxide molecules into a carbohydrate. And the carbohydrate, as I said, is usually described as being glucose, although it doesn't have to be. So one of the things that you're sometimes asked about is, what does it mean to fix something? Okay, you hear that term in chemistry and biochemistry in more than one place. You can do nitrogen fixation, you can do carbon fixation, you can even do phosphorus fixation. And what it means is anytime there is a form of a molecule that at one time is an inorganic substance, and it is transformed into being part of a larger molecule, where the larger molecule is an organic substance, that is a fixation. So why is it appropriate for what we're studying here? CO2, carbon dioxide, is one of the very few forms of carbon on Earth that is not considered to be organic. And these CO2 molecules, in combination with some water, are later going to be, or first part of a molecule that has this formula, C3H6O3. And if you take two of those, and bond those two together, even though this isn't showing how it happens, but what you will get out of those two is C6H12O6. So one of these two actually ends up being the direct product of the Calvin cycle, okay? It's a molecule that is called, abbreviated TP, or by name it's called triose phosphate. And when you see TP or triose phosphate, just automatically think half a glucose, okay? How does the name triose phosphate tell you that it is a carbohydrate? What's the clue within the name?
Os.
OSC. Glucose, sucrose, maltose. Virtually any, at least monosaccharide you can name, and the vast majority of the disaccharides you could ever name end in OSC. So triose phosphate is a carbohydrate. All carbohydrates are organic molecules. So this carbon is one of those three carbons right there. And that also tells you that you can't do this with one carbon dioxide. You'd have to have 3 carbon dioxides to make one of these. And you would need 6 carbon dioxides to make one of these. Okay, and that's a real common thing that's done in photosynthesis, is trace the carbons, okay? They might show you a simplified version of the Calvin cycle, and then say how many of this type of molecule, how many CO2s would you have to put in order to make 6 glucose molecules. And all you're doing is figure out how many carbons are in the 6 glucose molecules, 36, 6 times 6, and that tells you you'd have to put 6 CO2 in. Dana?
If it's called triose phosphate, is there a phosphate group attached to it?
Mm-hmm. There won't be when the two combine together into glucose, though. the phosphates that are attached to them will actually be the energy necessary to make the covalent bonds that will allow them to be bonded to each other. Because glucose doesn't have a phosphate, two phosphates within it. So what we need to do is go through the detailed reactions now that represent first the light-dependent reactions, and then secondly, once we get the ATP and the NADPH formed, we look at the reactions that represent the Calvin cycle, or the light-independent reactions. The best diagram I can give you showing the light-dependent reactions is where we're looking at just a very small area of a thylakoid membrane. So this thickness that I'm drawing from here to here represents the phospholipid bilayer of a thylakoid membrane. And to orient you a little bit in terms of the bigger chloroplast, this fluid in here would be the fluid inside the thylakoid. And therefore, this fluid would be the stroma. I thought sure you were going to say the fluid outside.
Does fluid inside the thylakoid have a name?
Yeah, intrathylakoid fluid. Okay, so one of the things that happens within the fluid inside the thylakoid is this is like any other fluid in a cell or part of a cell. This is primarily water. I mean, it's not pure water, but it's mostly water, just like cytoplasm and stroma is mostly water. And there is a pH environment, and there is an enzyme in this area that is very conducive to water molecules splitting. And I'm not talking about dissociating like water does normally into hydroxide ions and hydrogen ions. I'm talking about water just breaking its covalent bonds, okay? But it needs some energy to do that, and that energy is going to come from sunlight. This is not the only way sunlight is used, its energy, but it is one of the needed things that sunlight is used for. So at the right pH and with the presence of an enzyme to lower the activation energy, the water molecules split. And from any one water molecule, you get one oxygen atom, but nobody ever represents an oxygen atom just, well, I wouldn't say nobody. In inorganic chemistry, you would. But in this sort of thing, what you're interested in is where does the oxygen molecule come from? The molecule that represents the byproduct of photosynthesis. And so what you get is O2, because oxygen is O2. But in one water molecule, there isn't enough oxygen to do that. So you can either put a coefficient 2 here and show everything double, but what a lot of people do is say, this is enough oxygen to give you 1/2 of an oxygen molecule. And then you simply wait for another water to split, and then you'll get the other oxygen, and they will spontaneously join together into a diatomic oxygen. And this oxygen is the famous oxygen that simply will diffuse out. Eventually, it will diffuse out of the cell. Since this is primarily happening in a leaf, it's probably happening in a palisade cell of the leaf, the upper layer of leaves that are rich in chloroplast. and it will eventually leave the leaf through openings called
Stomata.
stomata. Okay? And that's where our atmospheric oxygen comes from. Does this plant need any of it?
No.
Yes.
Oh yeah, for
It does.
yeah.
Plants are aerobic. A lot of people fail to realize that. So the plant needs a little bit of it for its own cell respiration, but in good photosynthetic conditions, the rate of oxygen production by a plant far, far exceeds the rate of oxygen use for cell respiration. So most of the oxygen is given off into the atmosphere for other aerobic organisms to use. So what else do you get from this water molecule? You also get 2 hydrogen ions. And we'll use those a little later. Okay, I'm just going to let them sit there for a bit now, because for the plant, what is most needed are the 2 electrons that will be liberated from this reaction. Who remembers the name of this reaction? It has a name. Say again? No? Close though. Photolysis. It looks like photolysis. Okay, so this is representing the photolysis or photolysis of water. There was a famous guy, I never knew him, he never asked me over for dinner or anything, but he's still famous.
McGonegal's.
His last name was Hill. And some people will refer to this as the Hill reaction. I don't expect you to use that, but if you run across it, maybe in a multiple choice question or something like that, you'll at least know where it comes from. So, how and why does the plant need these 2 electrons? Well, when these 2 electrons are originally released, they're what would best be described as low energy electrons. Okay, they're not very useful, in other words, as they are. But embedded into these thylakoid membranes are big old huge proteins. And typically the surface of the protein that is towards the outside of the thylakoid, that surface of the protein is embedded with many, many, many pigment molecules. So the big old blob that I have drawn here represents a very large protein, and each of these smaller things that I'm now drawing represents pigment molecules that are embedded into the protein.
A protein.
How do you feel? How do you feel?
And this collection of molecules, one big protein and many, many smaller pigment molecules, is what's called a photosystem. And this first one is what is called photosystem 2. And it won't make sense why it's 2 when it's the first one that I'm talking about and the first one that's used, but I'll make it clear to you, and if I don't, remind me, because that's a pretty common thing asked about. Which photosystem does the electron first go to, and why is that the first one that it goes to, and what's the significance, and things like that. But I can't explain it to you until we get more built. Okay? So either one or both of these electrons becomes attached to any one pigment molecule. Okay? It's not a high energy electron, but it can become attached to a pigment. And what the pigment is going to do now is do what pigments are real good at, and that is accept light energy, and they do one of 2 things with that light energy. They either reflect it or they absorb it. And if it is a wavelength, a color of light or wavelength that is reflected, the reason we call the molecule a pigment is because it gives us that color. So the blue t-shirt back here is blue because that t-shirt has pigments embedded into it that when white light from these fluorescent lights strike it, those pigment molecules reflect our eyes blue light. But they absorb what?
Everything else.
Everything else. What is everything else? The red wavelengths of light, red, oranges, and yellows, in other words, and also the green in the middle. Okay, but something that is reflecting red, like the shirt up here, reddish, then that's a shirt that is reflecting red, but absorbing green, and also absorbing all of the blue wavelengths of light, blue, indigo, and violet. So for photosynthesis, the only wavelengths for any one pigment that are useful are whatever are absorbed. Anything reflected is useless, anything absorbed is useful. So for that reason, there isn't just one kind of pigment up here. There's a whole variety of pigments. Many of the pigments, I'm going to write them into here even though they belong up there. These are the typical collection of photosynthetic pigments that most plants use. They use one that's called chlorophyll, and then you'll see a small case letter A after it. It's called chlorophyll A. There's another one called chlorophyll, but there'll be a small case letter B after it. Both of those.
Eric Rosa, your parents is in the parking lot waiting on you. Eric Rosa, your parents in the parking lot waiting on you.
That's not my mom. Both of those reflect green and absorb other wavelengths of light, but it's a slightly different green. Anytime you say it reflects red or reflects green or whatever, you're generalizing, because any one pigment is real good at reflecting one exact wavelength, like 762, okay, nanometers of light. But if you go 763, 764, it's still pretty good at reflecting that wavelength, but not as good. So there's one wavelength that's perfect, and then diminishing returns when you go to the outside of that. So chlorophyll A and chlorophyll B both reflect greens, but a slightly different wavelength of green. In addition to that, there are pigments that collectively are called xanthophylls, and those reflect yellows and therefore absorb other things. And there's a group of them that some people call carotines, and some people call them carotenoids. And if you're describing them in a response, I don't think it matters which one you use, but you may see them in a question as either carotines or carotenoids. So I just wanted you to be familiar with both of them. So
What do those do?
those reflect reds and oranges.
So what's the purpose to this? Of those? Is it just like interesting, or is it like the carotenes and carotenoids?
You're jumping the gun here.
Oh, okay.
So in this cluster of pigments that are up here, the vast majority of them are chlorophyll A and B. But there's always going to be a few xanthophylls, there's always going to be a few carotenes, okay? And when this electron that's been freed up from the the photolysis of water, okay, this electron will simply bond to any one of the pigments, but it'll bond at a relatively low energy level. Okay, it'll be an electron that's in an orbital that electron is designed to be in. And then what that pigment will do is it will accept any one, that lambda symbol means a wavelength of light. It'll accept any one wavelength of light. And if that wavelength is one that it reflects, nothing happens. if it is a wavelength that it can absorb, it will use that absorbed energy to take that electron. Imagine this as the nucleus of one of the atoms of that pigment. And here is the electron that we've just added. There are other electrons there, but that's the electron we've added. And with that addition of light, this electron simply goes to a higher energy level. It jumps to a higher energy level. And now you refer to it in one of two ways. You can now say that it's either a boosted electron, or you can say it's a high energy electron. But one boost isn't enough, okay? There's too much energy necessary to make an ATP or an NADPH to just boost the electron once. So when this electron becomes boosted one time by one pigment, it is next passed to another pigment. Same electron, but it's now at the higher energy level. And that pigment accepts its wavelength of light. And again, if it's a wavelength that's reflected, nothing happens. If it's a wavelength that is absorbed, the electron, now part of a different atom, will now become boosted to an even higher energy level. And this probably will happen depending on which cluster of pigments it's in. This will happen 4, maybe 5 times, until eventually this electron gets sent to one particular pigment molecule. And it's always a chlorophyll A molecule. And it is always most sensitive to wavelengths of light where it needs a photon of light, that's where the P comes from, of exactly 680 nanometers. That's what that pigment needs, that chlorophyll A molecule. It's called the reaction center of the cluster of pigments. And all of the boosting of the electrons that occurred up to that point was for nothing but to get that electron attached to that P680 chlorophyll A molecule. And when that P680 chlorophyll A molecule gets struck by 680 nanometer wavelength of light, it is now enough of a boost. It is the final boost of the electron that the electron actually leaves the cluster of pigments and gets picked up by another protein. So here now is our high energy electron, our boosted electron. And it gets passed to another electron. And when it does that, each of these steps, it is basically coming down an energy level. So it's losing one energy level here, just being passed to this protein. It's losing another energy level here. But there's some good that's being done. The good that's being done is any hydrogen ions that are out in the stroma are now fair game to be drawn into the interior of the thylakoid. So these hydrogen ions are being drawn through the membrane, and they're beginning to accumulate inside the thylakoid. If this happens a lot, just electron after electron after electron, what do you think happens to the pH of the thylakoid environment?
Decreases.
Decreases, goes down. And I don't know exactly how this can be done, but they have tested the thylakoid fluid, and there is approximately a 10-fold increase of hydrogen ions here as opposed to out here. How much of a difference in pH is that?
Only one.
One pH number. When you go, oh, big deal. But one pH number is a 10-fold increase. We have 10 times more hydrogen ions in this fluid than we have in this fluid. So what we've created is a way to use those hydrogen ions, but not yet. Okay, we're going to use them momentarily. Before we finish this, we're going to look at what happens to this electron, because it's got to go somewhere. If it doesn't go somewhere, if it sits right there, then the next electron can't go to here. And the pigments that are boosting electrons here can't send their electron here. So it's just like in cell respiration. Something has to happen to the electron. You got to keep them moving, or otherwise everything backs up. So there is a second big protein with a second collection of
Me too.
pigments. And this second big protein is called, in its pigments, is called Photosystem 1. And it has a dominant chlorophyll A molecule at its center, and it is called a P700 chlorophyll A. And what this electron will do, once it gets to this point and has been used to bring hydrogen ions in, this electron will become attached to one of the other pigments, one of the other chlorophyll Bs or xanthophylls or whatever it is, and exactly the same thing is going to happen again. These pigments are struck by a particular wavelength of light. The electron becomes boosted again, and that electron will be passed to another pigment, just like it was before, and then perhaps to another pigment, just like it was before. Every time the pigment is receiving whatever wavelength of light it is able to absorb. And then finally this electron gets boosted to the reaction center, P700 molecule. So what does that P700 need?
Wavelength that it's going to absorb.
Wavelengths of light of exactly 700 nanometers. And when it does receive 700 nanometer wavelength light, it will boost that electron to a high enough energy level that it once again will be passed to another protein. But this time, we're not going to use it to draw in hydrogen ions. We're going to use this high energy electron to directly make a molecule. And that molecule is going to be NADP plus hydrogen ions plus these high energy electrons. And that's where NADPH is going to come from. One of the two molecules we wanted to make. Remember, the whole point of this set of reactions was to make ATP and NADPH. So the same electron that we started with over here from water and boosted it, used it to bring in hydrogen ions, and now we've reboosted it. It now becomes an electron that is holding that hydrogen onto this NADP molecule and represents stored chemical energy. But what haven't we made yet? ATP. And ATP is going to be made, oh Lord, I didn't leave any room. We're going to make it over here. You make it anywhere on your membrane where you have room, okay? Draw more membrane if you have. Okay, so right here is going to be a channel. And this isn't very well shaped for what it really looks like or whatever, but it's just a cartoon version. And it's a hydrogen ion channel. Can hydrogen ions freely move through a membrane?
No,
Why?
because they're not.
They're polar. They're really, really, really small, but they're polar and they don't fit through. This is a trick to get them through by using electrons to pull them through the polar membrane, but that's a trick, okay? They just won't move through on their own, okay? But if you create a hydrogen ion channel where the interior is polar, and you make the channel really, really small on the inside so other polar substances can't go through it, then hydrogen ions will go through the channel simply following their concentration gradient. So where are they highly concentrated?
On the inside.
On the inside. We've accumulated 10 times more hydrogen ions on the inside, so they basically flow through this channel. And always flowing in this direction. And on the other side of this channel, maybe a part of the channel itself, there is another protein called ATP synthase. And it catalyzes the reaction where ADP and a inorganic phosphate group will use the energy, sort of the kinetic energy of the flowing hydrogen ions. The enzyme will lower the activation energy necessary to do this, and these two are joined together into ATP molecules. And that accomplishes our other thing that need to happen during photosynthesis. We now have a way of getting NADPH, and we have a way of getting ATP.
So that's, ATP synthase is just an enzyme that catalyzes this reaction?
Yes. Otherwise, the hydrogen ions flowing through the channel would not be enough energy without the enzyme being there. During this reaction, are electrons, do they constantly need to be resupplied, or are they recycled?
Recycled. They're recycled.
Yes and no. They're recycled in that they never go away. They're still in the general environment, but they're not going to stay here, okay? Because where is the electron right now? It's here, okay, holding that hydrogen onto NADP. And the NADPH is going to go over to where the Calvin cycle is occurring, and that electron is very unlikely to come back here, ever, okay? So people call this non-cyclic photosynthesis. And the non-cycling is the electron, in that the electrons have to be constantly renewed. And where do they come from? They always come from water, okay? And water is virtually limitless for this process. So it's not like there's a big problem in reforming them. But there was a stage of photosynthesis, an evolutionary history, so to speak, of photosynthesis, where the biochemistry was similar to this, but not exactly the same. And it was what is called cyclic photosynthesis. And what was being recycled were the electrons. And if you recycle electrons, what can't you make? NADPH. So in the evolutionary history of plants using photosynthesis, the original molecule produced by the light-dependent reactions was ATP only, no NADPH. And what that version of photosynthesis did, the easiest way to explain it, was it used this photosystem coupled with that electron transport chain. This didn't exist. The NADPH didn't exist, or any part of it, okay? But it did bring in hydrogen ions, and it did have hydrogen ions flow through to make ATP. It's just one electron over and over again. I mean, there are probably millions doing this at the same time, but one electron would start somewhere and be boosted and boosted again and boosted again, and then boosted by P700. And rather than going here, because that didn't exist, it would go here. This one was located next to this one. And then when it went here, that would help bring in hydrogen ions. And when the electron went here, that would help bring in hydrogen ions. And then the electron would come right back here and do it again, and again, and again. And that's why it's called cyclic photosynthesis. It produced ATP only, and the Calvin cycle was redesigned to use ATP only. Does that sound like it's as efficient as this? No, it wasn't. But all biochemical pathways have a history to them, have a biochemical history to them. And that, the reason I told you that, is that's why this is called Photosystem 1. It was the first one to exist. Discover, exist, evolve, however you want to think about it. There was a time period when that cluster of pigments in this protein existed, but that one didn't. But when this one came into being, and who knows how it came into being, in the sequence of how it was being used, it was used first, and this one was used second. But it explains why that one's called Photosystem 2 and this one's called Photosystem 1. There are a few, very few plants on Earth that are considered to be very evolutionarily rudimentary, like some animals are, rudimentary animals that haven't changed much in evolution as much as others. And there are a few plants that use cyclic photosynthesis even today. Okay. Not many though, because it's not a very efficient process. Before I get rid of this diagram, because we need to do the Calvin cycle, is there any other questions on it? Because it'll be much easier to answer questions when the diagram's still there. Julie?
The hydrogen ions, do they, when they go through the channel, just stay back in the stroma until an electron moves in there?
Yeah, if you think about what's probably going to happen to them, they go through the channel, be used as kinetic energy for making ATP, and then they become part of the hydrogen ion set that's out here. But then if this is daytime, there's going to be more electrons that are drawing them back in. So the hydrogen ions are recycled. Okay.
If you don't know, it doesn't matter. That's how it works.
Yeah, there's an ATP synthase.
So it goes into that protein, connects it with the other phosphorus.
Yeah. Using the kinetic energy of the hydrogen ions. Because they're flowing.
I just want to know where it happens. I don't understand what kinetic energy is.
So does this affect the immune system?
I dare you, I dare you. Ask him.
Oh my god.
Jeremy, ask him.
Sure.
So Mr. McGonegal,
The nervous system. Because we get nervous.
Mr. McGonegal. What should I say? Immune system.
Nervous. Because we're doing it.
Hey, name himself. I hope he gets it. I hope he doesn't just like you know what I'm saying?
Just like a wink of.
Electron carriers, they're part of an electron transport chain. You see it in books, it's like EPC.
Bro.
Ready?
Yeah, you can name all of them, but I don't think anybody's going to see that.
interesting. So how does this exactly affect the nervous system?
Well, for some people, it can dull you out and put you to sleep.
Yeah. Wait, doesn't it take 2 electrons?
Like there's somebody back there with a raid shirt on that it did that to them. And, I don't think anybody else that I saw.
Well, doesn't that take 2 electrons? ADPH. I know.
Take over for Briggs.
All right, so the Calvin cycle, or the light independent reactions, will not take as long. It's not as involved, but it is even more confusing. Okay? So try to stay with it, and it's something where what you got to remember is when we are studying reactions like this, whether it be the Krebs cycle in cell respiration or the Calvin cycle in photosynthesis, we're looking at just a person's representation of it. It really is nothing more than a bunch of molecules, some reactants, some enzymes, etc., that are colliding with each other in random ways, and we can't understand it that way. Okay, we can't draw it out where we say here's a random molecule and a random molecule, and these two happen to collide, etc. So what we show is a perfect representation of it. We show only reactions that are fruitful, only reactions that actually do lead to a product, and we show them as if this is like, this happens all the time. Well, it really only happens in probably 1 out of 1,000 or more molecular collisions, but these are the only collisions that lead to anything useful that is occurring. In the stroma, this is where CO2 is diffusing into. Where's it coming from? It has entered into, through an open stomate, typically in the bottom of a leaf. It's gone into the air spaces of the spongy cells of the thickness of a leaf. And now, odds are, it's now diffusing into a palisade cell, which is where most of the photosynthesis occurs. And even though you may think this makes it more complicated, I think it's easier to understand. We follow 3 carbon dioxides at a time rather than 1. If I show you 1 carbon dioxide at a time, then I'm going to have to show you 3 sets of reactions. If I show you 3 carbon dioxides at a time, then I can show you 1 set of reaction, and what you simply have to remember is that's 3 different reactions in some places. And I think that will make sense to you. Within the stroma are pre-existing molecules that are called ribulose bi- or diphosphate. I don't know why people don't get together and decide, is that going to be a D or is that going to be a B? But I see it in different references both ways continuously. So by abbreviation, this would either be RUBP or RUDP. And there's no difference in them, it's just a nomenclature difference. And those molecules that are RUBP molecules are 5 carbon molecules. Where do they come from? They're always regenerated, okay? So by the time we finish, we're going to have to make those 5 molecules again. Otherwise, the Calvin cycle won't be a cycle. Well, if we bring in 3 carbon dioxides, what we're really bringing into the reaction is 3 inorganic carbons. And 1 reaction at a time. Okay, 1 carbon dioxide at a time. Let me not do it this way. Let's do it this way. We're going to take 3 of these 3 ribulose biphosphates, which represent 3 5-carbon molecules. Each of them is a 5-carbon molecule. And we're going to add to them 3 1-carbon molecules. What are the 1-carbon molecules?
Carbon.
Carbon dioxide. What are the 5-carbon molecules?
Ribulose biphosphate.
How many of them are there?
3.
So if we add one at a time, one of these 5 carbons to one of these 1 carbons, we'll do a reaction in which we make a 6-carbon molecule. And then we do it a second time, because there's a second carbon dioxide, and there's a second 5-carbon RUBP. And we get 2 of these. And then we use our third one, third carbon dioxide to third RUBP. So by the time we're done, we've made 3 6-carbon molecules. And I don't know why, but those are the most ignored 6-carbon molecules in nature. I don't think I've ever seen a reference that ever gives their name. People will say they're ephemeral, okay? It's almost like they're, I don't know, we don't think they're important, okay? So I don't even know what their name is. I've never seen their name. But what they'll do, these 3 6-carbon molecules, is they will immediately split in half. Each immediately splits in half. So Joey, how many of them are there going to be?
6.
And how many will each carbon have?
3. Each chain.
We'll end up with 6 3-carbon molecules. And those molecules do have a name. Been a year.
Yeah,
GP, glycerate 3-phosphate, G3P is what they're abbreviated.
Yeah, G3P.
And I know you all have not had a tremendous amount of organic chemistry, but many of you have had enough that there are clues within molecules that you can use to help you remember what some of them are. Okay, so for example, is there anything in this molecule that can help you remember the fact that it is a 3-carbon molecule? Yes, there is.
No.
The prefix on the first word.
Bliss.
will help me.
So what other molecule do you know of that is
Glycerol.
something you're expected to know that has 3 carbons?
Glycerol.
Okay. Does anybody know what the 8 at the end of it indicates? I'm sorry?
Originate.
So it can also be called glyceric acid. And because it is an acid, the only organic acids either have phosphate groups or they have carboxyl groups. Okay? And this one has a carboxyl group, but it also has a phosphate group. And that's why they couldn't just call it glyceric acid or glycerate. They had to name it glycerate 3-phosphate. Do you think there's 3 phosphates?
Hell yeah.
No, there's 1. The 3 tells you where it is. What's the only other part of this molecule that has anything that's tangible, important? The carboxyl group. So the carboxyl group is on carbon 1, the phosphate group is the other terminal carbon. Do you need to know any of those things? No. But the more you look at a name and you go, that thing makes a little bit of sense to me. It'll be something that allows you to hold on to it better. It'll be something that allows you to understand it a little bit better. Okay? So, how will you see this if it's abbreviated? You'll see it as G3P, glycerate 3-phosphate. And there will not be one of them, there will be 6 of them as part of the Calvin cycle, as long as you put in 3 carbon dioxides. And in the next reaction, all that happens is there is a functional group change.
It's a rearrangement enzyme.
And what you end up with are the TP molecules that I told you about earlier. Triose, phosphate. So they're no longer giving you a number. Why do you think they're no longer giving you a number?
Because it's tri.
No.
Os 3 sugar.
It has 3 carbons and a sugar, that helps.
Oh, because
But what went away?
the carboxyl.
Carboxyl. Triose is a sugar. Do sugars have carboxyl groups? Does glucose have a carboxyl group? Does sucrose? Does galactose? None of them do. So the functional group change was to get rid of the carboxyl group here. It turned the molecule into a sugar, nothing but carbons, hydrogens, and alcohol groups, but there still is a phosphate group attached to it. Okay? And how many of these will there be?
6.
6 of them. What are they abbreviated?
TP.
TPs. Triose phosphate. What did I tell you the useful product of the Calvin cycle was going to be? Triose phosphate. So out of these 6, 1 TP
Why is it up here then?
is
Why is it
basically kicked out of the party. Okay? You go do something else. The remaining 5
Oh my god, you gotta be kidding me. It's gonna be the ugliest diagram ever.
stay in the cycle. So here's 5 TP right here. There's the 6th one right there. These 5 TP represent how many total carbons?
15.
15 carbons. How many total carbons do we have right here? 15. Amazing. And in the last reaction of the Calvin cycle, these 5 TPs are reorganized into a new carbon backbone, into these 15 carbons. 15 carbons becomes 15 carbons. 5 molecules becomes 3 molecules. So what's the only good we got out of this?
1 triphosphate.
1 triose phosphate. There it is. All of this, and you were in here for over an hour, and what you got out of it was 1 TP. Okay? But that's not the way plants look at it. They look at this as a factory. Okay? They say, I'm not going to send you 3 carbon dioxides into the stroma. I'm going to send you 3. And then 3 becomes 3 million, and then 3 million becomes 9 million. And pretty soon you start accumulating lots of TP in the stroma. And how many of them do you need to add together to get a glucose molecule?
2.
2 of them. How many do you need how many glucose molecules do you need to add together to make a starch molecule?
300.
Hundreds to maybe even thousands of them. And then all of a sudden you start seeing how this chloroplast is a food factory, okay? All because TP is being made and converted into glucose and then starch. Yes?
Do we need to know more specifically the ATP and the NADP?
All right, that's the next thing we were gonna talk about, and yes and no, I'll give you a version of it, okay?
So would you need 6 carbon dioxide molecules to make 1 glucose?
Yes, you would, because what you'd have to do is you'd have to put in 3 carbon dioxide to make 1 TP, then put in another 3 carbon dioxide to make a second TP.
So 2 rounds of the
2 rounds of the Calvin cycle. So, Arjun's question was, how do we tie these 2 things together? Well, some of these reactions of the Calvin cycle are not free.
That's the glucose.
They're energy
I
demanding
know, but like when they go
reactions.
together.
So the first reaction that requires energy is this reaction. And this reaction, every time it happens, 6 these to 6 of these, every reaction requires an ATP and an NADPH. So in total, 6 ATP are required right here. And 6 NADPH are required right here. So that's a very, very energy demanding reaction. And it initially didn't look like it. Because what's happening in that reaction?
Or
Functional group change. But that functional group change apparently requires a lot of energy. The reaction that is also energy demanding is this one, the reorganization of the carbon backbone. But this one is not as energy demanding, it requires 3 ATP. Most questions that I've seen having to do with linking the light dependent reactions to the light independent reactions don't require that you know numbers like this. They simply are asking you to know that the ATP generated and the NADPH generated during the light reactions are used for 2 of the reactions of the Calvin cycle. Okay? Yes?
So, going back to the light dependent reactions, isn't it, I guess, plausible that there would be a lot more ATP produced in the time that you could produce one NADPH?
Possible.
Okay.
In fact, you need that because it's basically a 9-to-6 ratio of ATP to NADPH.
Is there like a specific ratio from hydrogen ions that are traveling from the thylakoid, the
No,
interthylakoid?
there's
Okay.
too many other factors
Okay.
that are affecting that. I'm going to tell you one more thing. I forgot what it is. I guess it doesn't matter. For AP students, you might, I'm not going to do this now, but what you might want to do is look up very briefly what's called C4 photosynthesis and CAM photosynthesis. And neither one of those changes what we're doing here, but these are plants that live in unusual growth conditions, typically very high lighting conditions and very moist conditions. And under those conditions, they can't leave their stomates open all for very long because they'll lose too much water. So C4 photosynthesis and CAM photosynthesis are little derivations of this that allow them to survive in unusual growing conditions. So you wouldn't have to know the details of them, but just know a little bit about them, okay? All right, please return any chairs to where they came from and sort of clean